Racemosus is a species of fungus in the microaceae family. It's commonly found in soil, decayed organic matter, and building. It's also reported in human and animal tissue as well. The significance of Mucor Racemosus in the environment is help to decompose organic matter and involved in nutrient cycling. Mucor Racemosus also utilized in the fermentation food process and used in the production of various enzymes. Mucor Racemosus cause allergy to and has effect exclusively in immunocompromised individuals. The symptom is sinusitis, rhinitis, and alveolitis. Mucor Racemosus was used for allergen medical testing to test the production of IgE antibody. Mucor Racemosus was classified in domain Eukaryota, kingdom Fungi, phylum Zygomycota, class Zygomyces, order Mucorales, family Mucoraceae, genus Mucor, species Racemosus.
Assalamualaikum and hi everyone. This is my part where I would like to share with you more about the morphology of Mucor Racemosus. Mucor Racemosus is typical of fungi and related to animals. Both are eukaryotes. As we know that eukaryotes have through cell membrane, through nucleus, and through organelles. They also have cell walls that consist of chitin, ergosterol, and beta-glucan. Next, here we have a picture of the structure of an example of a mucor. As we can see, the Mucor Racemosus has columella, sporangium, sporangiospores, and sporangiophores. Just like other Zygomycota fungi, columella are spherical which provide structural support to sporangium. Sporangium is a spherical structure that forms a tip of sporangiophore. And sporangiophore are specialized hyphal structure that bear the sporangia. Lastly, sporangiospores usually either erect, simple, or branched, and grow up to several centimeters in length. The color based on the University of Adelaide research, they found that the colony's color is white to yellow, then becoming grey with development of sporangia. That's all from me. That's all from me for this part. Thank you. With me again, in this section, I will explain more about the reproduction of Mucor Racemosus. As we know that fungi doing both sexual and sexual reproduction, the same as the Mucor Racemosus too. Here is a short video that I will explain based on it. Sexual reproduction followed by mating of haploid hyphae, then formation of gametagia. For each hyphae, which the hyphae is haploid now. Then they undergo plasmogamy, which is a fusion of protoplasm to form zygosporangia. Then follow with karyogamy. The formation of zygosporangia now is diploid because fusion of nucleus. Then they undergo meiosis of the development of zygospores from zygosporangia. The zygospores now are diploid and undergoes meiosis, produce sporangiophores which are haploid. Some of the sporangiospores germinate into the new mycelia. Next, we will continue with sexual reproduction, which is important to Mucor Racemosus and other Mucor for propagation species. Sexual reproduction is a continuation for a sexual reproduction earlier. Since the spores are already haploid, so they are undergoing mitosis to produce unicellular spores, which are identical to mycelium. Those spores then fall on the ground and produce more mycelium, and then undergo mitosis to produce another multicellular mycelium. The mycelia will grow and repeat sexual reproduction. The cycle will repeat again and again and again and again. looking back at its production is a cycle for Racemosus and other mucors to
move on to a nutritional mode of Racemosus. As we all know, mucor is an heterotroph because it cannot produce their own food due to the absence of a pigment in which involved in photosynthesis. Thus, it can't undergo a photosynthesis process. Racemosus is a common species found under a genus of Mucor. So it has a same nutritional mode with some other species under a mucus. So Racemosus undergoes a saprophytic lifestyle in which it obtains nutrients by decomposing dead and decaying organic materials. It also one other good decomposer that can be found, which an earlier decomposer too, that can be found by dead material or dead organism, in which they can utilize the ability, which is quick absorptions. They can they manage to absorb all the nutrients before other decomposers come. It also it undergoes a process called a nutrient breakdown, in which carbohydrate will break down into a lignin and a cellulose. So move on. Habitats of Racemosus, it usually found in the soil and a dead organism. As I mentioned before, Racemosus is a mucor, which is a heterotroph, so it depends on the external source of nutrients. So in the soil, as we know, soil contains many nutrients, so it is a place where Racemosus can farm or get a nutrient at its finest. Also, that organism, as I mentioned before, it is a saprophytic organism which gain nutrient from dead materials. So, Racemosus also is a dimorphic zygomyces, in which it has more than one appearance. It can be in the mycelial forms or in a yeast form. In the presence of air or aerobic conditions, it will form or develop a grey mycelium that will darken into black or brown sporangia, which contain sporangiums for the fertilizations for reproductions. So in the absence of air or in anaerobic environment, it tend to be a yeast forms. So high carbon dioxides also promotes the development of yeast forms. I forgot to mention about anaerobic environment. It also required a high concentration of hexose in order to produce yeast forms. Okay, thanks.
Racemosus is a fungus that has many impacts in this world. In this section, we are going to share the significance of these species in three major scopes, which are ecological, its uses in the biotechnological and industrial fields. The significance of Racemosus for the ecological is that this fungus is a good decomposer which can be beneficial in nutrient cycling, soil health, and microbial interactions. The Racemosus is known to be saprophytic fungi, where it can break down and decompose organic matter which keep the nutrient cycle for sustaining life on Earth. When they do the decomposition of the complex organic matter, they actually make it easier for the plants to grow by absorbing the nutrients which are the products that fungi release, like carbon, phosphorus, and nitrogen. The presence of this fungi is important to keep the carbon cycle and nitrogen cycle, which keep recycling these elements from the atmosphere to the soil, to the organism and back to the atmosphere. Moreover, it can also be good for the health of the soil, as the fungi decompose the organic material like leaves, wood, and branches that fall at the surface of the soil. It will produce humus, an organic compost that can improve the soil structure. Other than that, the fungi can also interact with other microorganisms in the soil, like bacteria and other fungi, that will help improve nutrient cycling and soil health. The other significance of Racemosus is its application in industrial fields. It is used for the production of various enzymes, such as lipase, protease, and amylase. Lipase is produced from mucor species, including Racemosus, which will be going through the fermentation process. Lipase was used in industrial fields to catalyze the hydrolysis of fats into glycerol and fatty acids. It will improve the texture and softness of the cheese, but it also acts as a flavor enhancement agent in butter and margarine. Lipase was not only used in food industry, but also used as the stain remover in detergents. These fungi also produce protease, which is an enzyme that breaks down proteins into peptides and amino acids. This enzyme was used for the process of tenderization of meat and cheese making. This enzyme is also important for students to conduct their laboratory experiments, and with the help of this fungi, protease will be easier to obtain. Other than that, Racemosus is also used in the production of antibiotics, since it can produce secondary metabolites with antimicrobial properties that naturally kill or inhibit the growth of bacteria. Even though most of the antibiotics are made from other species of fungi, Mucor species also have the potential for the new discovery of antibiotics.
In biotechnological fields, Racemosus is used in bioconversion process due to its enzymatic capabilities. Biofuels are one example of a bioconversion process which is used to produce ethanol. Racemosus can convert lignocellulosic biomass, which refers to plant dry matter, into fermentable sugars. The sugar will be fermented in the starches of grains such as barley and be fermented by yeast or other microorganisms to produce ethanol. Ethanol was also used in wheelchair when it was blended with gasoline. In the United States, nearly all motor gasoline is now sold with about 10% ethanol by volume, which is known as E10. It will boost the performance of your engines since it has a high oxygen content and burns more completely than ordinary gasoline. But only some wheelchair that could take gasoline with 15% ethanol, which is also known as E15, since it can degrade rubber and plastic in engines and fuel lines. Racemosus is also used in the production of lactic acid from lignocellulosic biomass, which is used in food preservation and biodegradable plastics.
Commercialized products of Racemosus is often used in a fermentation process of productions of seafood, which is a fermented soybean curd. There are many types of fermentation actually. For example, we have a natural fermentation method, bacterial fermentation method, enzyme ripen method, and lastly is a mold fermentation method. There are studies that was made to differentiate between these types of fermentation method, which involve natural fermentation method and lastly mold fermentation method. So from the research, it found that in natural fermentation method, the high degree of protein hydrolysis was occurs, and the total number of bacteria was found is high. While in a mold fermentation method, even though the low degree of protein hydrolysis, but the total number of bacteria found was lower. And for natural fermentation methods, the internal structures might be denser and uniform, while in mold fermentation method, even though it has a less denser internal structure, but it has a great adhesiveness and hardness of external structures. So both of the product produce 50 aroma compounds and safety to be consumed. So from all the results that I have seen, can be said that mold fermentation method is more better compared to natural fermentation methods.